Good evening, everyone. Hello, good evening, very good evening to all of you. Good evening, everyone. This is Vaishali from Spark Academy, and we will be going to discuss about plant physiology and its PYQs. And if you have any doubt, you can contact here. We can solve doubts, your doubts together. You can post your doubt in form of chat, and I'll be giving you answers. Yes, please, can I have some quick comments, please, if you can hear my voice and see the screen properly? Yes, good evening, everyone. Can I have some quick comments? Quick comments. In chat section, you can comment that if you can see the screen properly and if you can hear my voice clearly. Yes, good evening, Anjali. I hope you can hear my voice clearly, Anjali. Yes, hello, good evening, all of you. Once again, very good evening and a warm welcome to all of you. And this is Vaishali Kamdar from Spark Academy. And yes, last week I have taken session on, 4 months plan of NEET, how to crack NEET with good planning and targets, daily target. I hope you are achieving daily targets and thereby you'll be cracking NEET with good score. Yes, so along with that, you might require certain type of helps in few important chapters and important units. So we are going to discuss that today. One of that unit that is plant physiology. Yes, thank you so much, Anjali, for your quick comment. So we will be discussing previous year MCQs with solutions today. And yes, for more updates, you can follow us on following links. So sparkacademyonline.com, Instagram, Telegram, Facebook, anywhere you whatever is your convenient way, you can follow us on that. So this is what see I'm introducing my team with you. So physics, maths, and biology. That's me, Vaishali. I'm taking care of complete biology. So, if you join Spark Academy, what do you need to do for that? You just need to download this app from your Play Store, that is Spark Academy app. You can see the logo over here, the blue color logo, which is on left hand side corner. So you can select this logo from your Play Store and download our app, then download and then join any of the course which is convenient to you. Then you will be having daily live classes and in session doubt clarification also, and free materials. We are providing tips and tricks for Eamcet, J as well as NEET. So admissions are open right now for 2023 for classroom online coaching as well as short-term crash course and regular. So see, after downloading, yes, hello, good evening, Niharika. After downloading this app, you can use this coupon code that is VAI10. VAI10 Vaishali 10. So you will be getting a huge discount on all the courses offered by Spark Academy. Yes, everyone. And you will be enlisted in my list, and then will be directly contacting. And I'll see this is how you can get good huge discount also. So session plan and weightage, if we discuss, then yes, we'll be planning, and in this session we'll be discussing few basic concepts or few important concepts from plant physiology. That is first this unit, this important unit, and you if you see the weightage part of it, then one or two question, questions asked from each unit. Most probably respiration and photosynthesis include certain type of pathways which you need to understand. Yes. So I'm discussing concept right now, basic concept. To see, directly we are moving towards the important point of it. Just a second. Yes. So transport in plant. Transport in plants. So in case of transport, there are two types of transport basically. If it is long distance, if it is to long distance transport, then it is known as translocation. And in this case of translocation, what will be used? Xylem and phloem. You'll require involvement of xylem as well as phloem. So this type of transportation to the long distance, that is known as translocation. Yes. So then different means of transport we can discuss in this chapter, though active transportation, passive transportation, facilitated diffusion, simple diffusion, osmosis. All these basic concepts you will discuss in detail later on. But right now I am directly moving towards how see how it is going to, how water can be transported from root hair to xylem. So this is the one diagram given in your NCRT. So here you can see this is what a bypass route. Yes, you can see here this is what a darker part, that is a bypass route, or you can say it's apoplast way of transportation. So when water, see from if you look into the anatomy of roots, then what is there? First outermost root hair, which is actually protrusion of the epidermis layer. Yes, so epidermis layer is the single layer of epidermis. It is directly in contact with water. So what happens here? Water either directly enter into the cell, or it can use cell wall, this type of pathway. Okay, but this type of bypass route it can use up to this is what a bypass highway route. Okay, just like how we use bypass highway route, like that this apoplast is fast because it will be from cell wall to cell wall, cell wall to cell wall. So up to what point there is a checkpoint here, check post here, that is known as Casparian strip, which is there in the endodermis layer. You know that cells in endodermis layer, they have deposition of what? Yes, Niharika. Anjali, Niharika, those who have joined, I can see a lot of numbers here, but I can see two names only in my chat. So you can communicate in form of chat right now, guys. So I'm asking you, my question is in endodermis layer, cells present in endodermis layer. So which type of depositions they have, and especially in which part? Especially in which part they have deposition and of which material? Yes. Yes, Nitin, you can also answer. Endodermis layer cells present in endodermis layer, they are specialized cells, they have certain type of deposition. Which type of deposition? And that is completely complete deposition, or in angles, or in which part? Yes, no, Nitin, it's not Saberian strip, but it is known as Casparian strips. But yes, deposition of Saberian. Okay, deposition of waxy material Saberian. That's right, Nitin. And it is in which part they have this deposition? Niharika, angular deposition or completely forming double cell wall type of deposition? Yes, Rakshan, or I. So you can also answer, those who have joined. Come on. Which type of in which part they have deposition? Angular, completely, double cell wall formation, or at which part? What is this called? If a circle is there, then if you would draw a line just touching to the one point like this, then what is it called? This is this is called what? Yes, Anjali, can you tell me this thing? This is known as tangent. Okay. So deposition of some Saberian tangential line. Remembered or not? I hope you remembered. Epidermis, then cortex layer, several layers of cortex, then endodermis. This is what we are discussing about anatomy of flowering plants. I know this is not in plant physiology, but when they are asking any question in NEET, then you can't say, sir, this is not from plant physiology. This is actually comes under anatomy. Right now, when only 122 days left, or 122 or 123 days left, your concept should be clear. Yes, Horera. So this is what a tangential line of endodermis cells. They have seborrheic deposition of waxy material which can stop this movement in apoplast way. Okay, so this is how they'll be stopping this movement by apoplast, and then it will become symplast like this. Okay, what is symplast? In symplast, they are entering, water will enter into cytoplasmic membrane, and through cytoplasmic streaming, it will be passed on to next cell by plasmodesmata. So here plasmodesmata is required, and this is how it will travel till xylem. So always remember, final transport of water from a soil, here it is here soil if you assume, then from soil to xylem, through epidermis, cortex, and endodermis and pericycle, finally to the xylem, root xylem. It is final reached by symplast. Always finally it is by symplast. Yes, everyone. Is that clear? So always remember, but slow one is symplast, final one is also symplast, and fast is apoplast. You can use fast one. You can use fast one till endodermis, till Caspian strips. Yes, everyone. I hope it's clear. Now, after water is reaching to the xylem, root xylem, it will be pulled up till the any tip of the plant by transpiration pool and root pressure. So two things work here for transport of water or translocation of water till tip. So in that case, which 2 points will, it is known as transpiration pool model. Okay, adhesion, cohesion, transpiration pool model. In that, it will be because of pulling force. That is transpiration and pushing force. So modest push, not a complete push, but it's a modest push. So modest push is given by root pressure. Yes, everyone. So I can see a lot of people has joined this class. Yes, can anyone answer what is guttation? What is guttation? Yes, Vikram, Hurera, Rakshan, Nitin, Niharika, Anjali, and few more, those who have not answered anything. Yes, can anyone answer this? What is guttation? Guttation is because of transpiration or root pressure. Yes, I want this answer. Guttation is because of loss of water in liquid phase, right, Nitin? Loss of water in form of liquid, that is guttation. It is because of root pressure, not because of transpiration. Okay. In the night time, evaporation rate becomes low at that time. Transpiration is a type of evaporation only. So root is applying pressure, and because of that, water will ooze out, not from stomata. Remember, not from stomata. There are specialized structure. Yes, so that is because of that, root pressure is the result. Guttation is result of root pressure. So this is what we can discuss about water translocation. Then for final thing of this chapter, that is food transportation. So for that they have given us this diagram and NCRT. So this is what a diagram showing pressure flow, mass flow mechanism, or pressure flow, mass flow hypothesis. Pressure flow, mass flow hypothesis. So what happens here? This is source. You all know that green leaves their source of food because they can synthesize food by their on their own by photosynthesis. So food is synthesized in leaves. Okay, synthesized somewhere here. So this food is synthesized in form of glucose. Then it will be converted to what? Sucrose. Yes, everyone. Then sucrose is getting transported. This is what a short distance transportation to phloem. Okay, first it will be loaded in companion cell, then from companion cell, maintenance of regulation by regulating osmotic gradient, it will be translocated to phloem. So this is your phloem. So inside phloem, whatever dots you can see, they are sucrose molecule, for example. Yes. So if you think about a solution in which if you keep on adding more and more sucrose, then what happens? This solution becomes hypertonic or hypotonic. Yes. I want answer from everyone. So here in phloem, what happens? Solution inside phloem, it becomes hypertonic or hypotonic. Hypertonic or hypotonic. Yes, Surera, Nitin, Vikram. Come on, come on. What happens to the solution here inside the phloem near the source? Anjali, Niharika. Niharika, why I'm not getting any answer from you? Are you there? Yes, you're right, Vikram. So it becomes hypertonic. So here it becomes hypertonic. So phloem, yes, hypertonic in case of phloem. Okay. Phloem becomes hypertonic near source. We are talking about near source. Phloem becomes hypertonic and nearby what will be passing? Nearby vessel will contain xylem. Will be xylem. Xylem contain water. So water will get transport from from its own higher concentration to lower concentration. So here see, because of that, inside the phloem, what will be there along with that sucrose? You can see lot of water molecule also will be traveling. It will be transported from nearby xylem. Yes, everyone. So here what happens? Here pressure is created because of so many. So traffic is there. Too much pressure is there. Too much congestion is there. Yes, everyone. So in this region, it becomes congested or it becomes a pressure is created. So whenever molecules are under pressure, they'll move in nearby area in whatever region they feel low pressure. So here there are two types of sink they can travel. So either growing part of the tip or growing stem or root where they want to store this food. Yes, everyone. So this is how slowly, step by step, because of the pressure and because of the releasing of the pressure, they'll be traveling till sink. What is the, see, this tip of stem and root, both of them are acting as sink. Yes, everyone. This can be also sink, this can be also sink. Is that clear? Then if food is now food or sucrose, it has been reached till sink, then what happens? It will ooze out from, I mean, it will be transported, not oozing out, word we can use, yes, it will be actively transported from phloem outside in the sink. Okay, so what happens right now in phloem? It becomes, here phloem becomes hypotonic. Phloem near sink becomes hypotonic. So because it becomes hypotonic, water will be removed. Whatever water has been, I mean, it has been entered from xylem to phloem. Now it has been, it returned to the xylem again. So this is what happens in pressure flow, mass flow mechanism. I hope there is no doubt. If you have any doubt, you can ask me in chat section. So this is about transport in plant. Two main important points I have covered: biological nitrogen fixation. Now it is there, and mineral nutrition. So your reduction of nitrogen, atmospheric nitrogen, occurs by nitrogenase enzyme to ammonia. First fixation product, it is ammonia. So free living organism like Azotobacter, Bacillus, Bergeringia, Nostoc, and Abina, they can fix this the nitrogen. And symbiosis, Rhizobium and Frankia. Rhizobium is leguminous symbiont, and Frankia is non-leguminous symbiont. So leg hemoglobin is required for this. Okay, that is scavenger of oxygen here, because nitrogenase enzyme can be functional only in anaerobic condition, but these type of organism, in case of rhizobium, they are aerobic organism. When they are free living, they will act as an aerobic organism. So they have this leg hemoglobin, which will act as a scavenger of oxygen. So they will eat all the oxygen and then make the environment more comfortable, more favorable for action of nitrogenase. Yes, so this will require 8 electron, 8 hydrogen ion and 16 ATP for conversion of nitrogen molecule to 2 molecules of ammonia. So NH3 is formed, is used to make glutamic acid and transamination processes. And if we look into the metabolism of nitrogen, then atmospheric nitrogen, it will be fixed either biologically, industrially, or electrical nitrogen fixation. Then denitrification, then fixed in form of ammonia. Ammonia will be converted to nitrate and nitrite. So that is known as nitrification. So plant can uptake only nitrogen in form of nitrates. So they'll uptake this nitrates, and then a plant will be eaten up by animals and both will undergo decay, and this will result into ammonification, release of ammonia. And in some cases nitrate is converted back to atmospheric nitrogen, that is known as denitrification. Yes, do not go anywhere. I'll be discussing soon. I will start discussion of PYQs. So then you all know about glycolysis in respiration, where glucose will be converted to 2 molecules of pyruvic acid. This is known as EMP pathway. This is anaerobic process which occurs both in aerobic, anaerobic, all type of organism. This is common amongst all the organisms. Then fermentation, in which occurs mostly in muscle cell and yeast, where pyruvate is converted to either lactic acid or ethanol. In lactic acid fermentation, carbon dioxide is not one of the product. See, yes, Niharika, this decay, that is mineralization, that is known as mineralization. Some fungus may act upon it, some fungus may act up, they will act upon these animals and plants when they are died, and they'll undergo decay. Yes, Niharika, that is known as ammonification. So this is known as Mineralization, also it's known as mineralization. This type of decomposers, they are acting here and they'll convert microbes related decomposition. Yes, Niharika? So decay with the help of microorganisms. Okay, now in case of glycolysis, we were discussing where exactly, what is the location of glycolysis. Yes, come on, answer in your chat section. You're welcome, Niharika. Now, location of glycolysis. Come on, fast. Yes, Nitin, Vikram, um, Anjali, Niharika. Location of glycolysis. Everywhere glycolysis occurs in cytoplasm only. Okay, location is cytoplasm. So pyruvate will be formed in cytoplasm itself. Then fate of pyruvate, either it can enter to lactic acid for fermentation or it may enter to anaerobic condition, in that it may ferment to ethanol and carbon dioxide. But if not, if enough oxygen is present, then it may enter to aerobic respiration. This is a normal condition what happens in our case, or all aerobic organisms, all aerobic eukaryotes. Yes, Niharika, that's right. So pyruvate formed in cytosol, so enters mitochondrial matrix now. So this matrix, what happens in matrix? TCA cycle. So after TCA cycle, we'll be having NADH plus H plus and one substrate level phosphorylation occurs during this process. So ATP is also formed. So you will be having NADH plus H plus, and what is role of this NADH plus H plus? This will undergo oxidative phosphorylation, or you can say ETC. It will undergo in ETC. It will give you more ATPs. Yes, this is how ETS or electron transport system, electron transport chain occurs. So here you can see that this is what NADH plus H plus, which has been synthesized in mitochondrial matrix. So mitochondrial membrane, inner mitochondrial membrane contains all these type of cytochromes and all the carriers, all enzymes responsible for ETS. Yes, I hope oxidation of NADH plus H plus by NADH dehydrogenase, which is a complex number 1. Electrons are getting transported from FMN to FES, and that is complex number 1, and finally to ubiquinone. And ubiquinone will be converted to ubiquinol. This is what ubiquinol is. This is what a ubiquinol. And the same ubiquinol will receive electrons from complex number 3. Complex number 3. No, sorry, from complex number 2. From by succinate dehydrogenase enzyme. Okay, complex number 2. From here, the same ubiquinone will receive this electrons by this one. Then electrons are from ubiquinol to electrons are transferred to cytochrome B, then cytochrome C1, and finally cytochrome C. Cytochrome C, you can see here, it's a mobile carrier. Mobile carrier attached to outer surface of inner mitochondrial membrane. So electrons are then transferred from cytochrome C to cytochrome A and then cytochrome A3 and to copper centers. So these transfers coupled with the ATP synthesis, one NADH gives you 3 ATP and one FADH2 gives you 2 ATP. So then in photosynthesis, there are two types of phosphorylation, non-cyclic one and cyclic one. In non-cyclic one, two photosystems takes part, LHC1, photosystem P700 and P680. So first it is P680. This is PS2. Just a second. PS2. So it starts with PS2, that is P680. It will absorb light and then electrons get excited here, and this excited electrons will travel to travel in the farther orbit from the nucleus, and they will be accepted by certain electron acceptor. These are primary electron acceptor, and then they'll be transported to ETS and finally to PS1. So here, and again they are getting excited and then accepted by electron acceptor, and finally to NADH dehydrogenase. This is how you can observe this type of Z type of scheme. So this is also known as Z scheme. Then chemoosmotic hypothesis, how ATP is getting synthesized in chloroplast, because ATP is normally synthesized in mitochondria during respiration. But this is what hypothesis which shows that ATP can be synthesized because of this proton gradient created in thylakoid membrane during this transport of electron in photosystem 1 from in photosystem 2 to cytochromes. Okay, then Kelvin cycle. So how these carbon dioxide are getting fixed and resulting into formation of sucrose. So RUBP is a 5 carbon compound which will accept carbon dioxide and which will result into carboxylation, and it will be converted to 2 molecules of 3-PGA. So that's why this is known as C3 cycle also, because here first product form, that is 3 carbon containing compound 2-PGA, phosphoglyceric acid, which will undergo reduction, and then there will be production of ADP, there will be production of sucrose. So C4 pathway mostly occurs in C4 plants with Krebs anatomy. Those are having Krebs anatomy. So in that complete, the bundle sheet cells. So bundle sheet cells they are rich in rubisco, but mesophilic cells will be not having rubisco. They will be having PPKs. So PP is primary carbon dioxide acceptor in case of C4 plants. Yes, everyone, this is what we concept should be clear. If you have any doubt, you can ask me. Otherwise, we'll start PYQs. Admissions are open right now. You can contact number flashing below. Okay, the link of Spark Store is given in description box. So whatever course you want to join, you can join and you can buy it from your Spark Store. Do not forget to use VAI10, that is Vaishali 10 coupon code for opting for online coaching for any type of offline, online, any type of courses. So yes, first question. Are you there, all of you? Those who have joined, I can see a lot of people has joined. Yes, which of the following criteria does not pertain to facilitated transport? Facilitated transport. Yes, I want answer with question number, everyone. 1, A, B, C, D. You can write in your chat section. Yes, Niharika, Nitin, Vikram. I'm waiting for your answer, guys. This is very easy question, so that's why I kept it first. And this still it is easy. It was asked in 2013. Yes, can anyone answer, please? Which of the following criteria do not pertain to facilitated transportation? Facilitated transport. This is from transport and plant. So you might be knowing that For example, if I want to draw a triangle of concentration, then this is a peak where which is known as a high concentration point, and here the concentration is low. Okay. So if anything which is transported from its high concentration to low concentration, that is known as downhill process. Downhill. Anything which is transported in this direction, that is known as uphill process. So in which when you are traveling uphill, right, at that time you will require ATP, you will require energy. So here facilitated transport, it is a type of diffusion only. So type of diffusion in which what occurs, see this is a type of diffusion in which it is highly selective because proteins are involved here. Proteins are involved because anything which you need to transport with hydrophilic moiety. For example, this is a substance which is hydrophilic and you want to transport this across the membrane which is made up of lipid bilayer like this. You know that lipid bilayer contains hydrophilic heads and hydrophobic tails. So this is what a hydrophobic area. Hydrophobic area. So this hydrophilic moiety substance cannot travel directly through this membrane. So it requires help from membrane protein, integral protein. From that it can be transported. It can make this protein can make channel into it, or it can twist and turn and then transfer this. So this is why proteins are involved here. So proteins are very specific. So yes, it's highly selective and your transport also gets saturated. So, and it is not uphill transport. So this is what not criteria, not pertain to facilitated diffusion. That's why our correct option is, we need to select wrong one here. That's why correct option here is uphill transport. Yes, next question. We'll move on to the next question. Transpiration and root pressure cause water to rise in plant. As I have explained, one is pulling force and one is pushing force. So that's what you need to select here. So transpiration is what? Push or pull? It's a pulling. And root pressure, it's a modest push. So it's a pushing. So correct option here is option number A. The next question. A column of water within a xylem vessel of tall trees does not break under its weight because of, why it will not break off? See, water molecules, they have tensile strength. They are holding their hands together. If you think like that, then you will understand in a better way. They will never leave the hands of their neighbor or their friends. So that is what we call as tensile strength, and because of that only there will be never, see, if you suck anything from a straw, okay, this is a glass and that, for example, water is there, you are sucking this using the straw, so water will be coming like this. There will be no breaking in this column. There will be no breaking in the column because of the tensile strength of water. Then root plays an insignificant role in absorption of water, in which case? So root plays where they are not absorbing water, where already water is abundantly present. That means when in case of hydrophytes. So which of these are hydrophytes? Which of these is hydrophyte? Yes, here that's Pistia. So because it is hydrophyte, its roots are playing a significant role in absorption. Next is water potential of pure water. Remember, water potential of pure water, that is side W, is always 0, which is maximum also. Maximum. But this is under standard temperature and pressure. So STP condition without any pressure. Without any pressure. Without any pressure, water potential of pure water is 0. So if you think about this, then tell me what will be the water potential of solutions? Water potential of in solutions. Water potential is equal to what? Option number A, 0. Option B, uh, positive figure or more than 0. Or C, less than 0. Yes, can anyone answer for this question? Yes, I can't see who has joined right now because no, right now if you answer, then only I can call out your name. Solutions, for solution, water potential becomes what? So post this doubt in form of post this answer in form of question number 6. Okay, this could be your homework question. Sixth question, ma'am, for sixth question, answer is A, B, C, D. Post like that. Yes, I'm not waiting right now for your answers, guys. You can post your doubts if you have. The first stable product of fixation of atmospheric nitrogen in leguminous plant, it is always ammonia. We have discussed already. N2 will be converted by nitrogenous enzyme. By nitrogenous enzyme, it will be converted to first fixed product, that is ammonia. Then deficiency symptom of nitrogen and potassium are visible first always in as nitrogen and potassium. These two are substances or elements which can be remobilized. Can be easily remobilized. Yes, if these substances, these elements, they can be easily remobilized. That means when they are reaching to the senescent level, then they can transfer this elements to the growing tip. That's why first deficiency symptoms of these will be this will be observed in case of senescent leaves. Then, in which of the following all three are macronutrients? Macronutrients we need to select. So nitrogen, calcium and phosphorus, they are macronutrients. Then bundle sheath cells. Already if you have carefully and concentratedly attended this first half an hour of this class, then you might be getting all the answer correct. Yes, bundle sheath cells, they are rich in rubisco. They lack PPKs. Yes, answer is 8A. Now you're back, Niharika. Yes, 8A, they are rich in rubisco. So PPKs present in mesophilic cell. Okay, but in mesophilic cell, especially in case of C4 plants, in C4 plants, mesophilic cell lack rubisco, so that's why bundle sheath cells, they are rich in PPKs there. Sorry, they are rich in rubisco there. Next, yes, Niharika, as you are back, the structure that are formed by stacking of organized flattened membranous sac in cytoplasm. Niharika, this is question number 10. In chloroplast, flattened membranous structure in chloroplast is easy peasy. Those who have joined, they can answer. It's very easy question. Even 7th standard or sorry 8th 9th standard वाले भी answer दे देंगे. Yes, come on. Structure that are formed by stacking organized. See, what is this called, Niharika? And those who have joined, see, in case of chloroplast, if I draw structure of chloroplast, it will be like this. Spare me for bad drawing also, okay? I just wanted to show this. We are talking about this part of chloroplast. What are these called? Flattened membranous sacs in chloroplast. Stacked and organized. A. Grana, B. Stroma lamellae, C. Stroma, and D. crusty. Come on, quick, quick. We have only 12 minutes left. See, chloroplast is double membrane bound structure. In that, you'll find this type of stackled structure like this. And here you'll find a fluid matrix. So this is what we call it as granum. One single is granum. All membrane bound sacs in the chloroplast, they are known as grana. Yes. Next question. With reference to the factors affecting the rate of photosynthesis, which of the following statement is not correct? Okay, here question number I should change. So this is 11th question. Now you can answer 11A, B, C, D. Yes, increasing atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. C3 plants respond to higher temperature with enhanced photosynthesis, while C4 plant have more lower temperature optimum. So incorrect we need to find out here. So incorrect option is this. This is vice versa. C3 plants do not have higher temperature. They do not have higher temperature optima. C4 plants have higher temperature optima. Yes, next question is cytochromes. This can be as a 12th question. Sorry. So cytochromes are found in inner mitochondrial membrane. That means crusty of mitochondria. Then which of the metabolites is common to respiration mediated breakdown of fats and carbohydrates and protein? So common intermediate in all these pathways. Yes, yes. Can anyone answer? Those who were joined. You were answering concept-based question, but why not this MCQs, PYQs? This class is for that. Yes, please answer everyone, so that I can come up with more questions and more these type of sessions where we can discuss only question answer. Yes, Nitin, Vikram, Rakshan. Niharika, come on. Yes, this is 13th question. So 13C is correct answer. That is acetyl CoA. Acetyl CoA is common intermediate between all of these. Then which one? This is 14th one. Spare me for these numbers, guys. 14th one. So in which one of the following processes CO2 is not released? I've already explained why CO2, in which of these is CO2 not. See, in case of fermentation, alcohol fermentation, one of the product along with ethanol is carbon dioxide. Aerobic respiration in animals, aerobic respiration in plants, wherever it occurs, carbon dioxide it results into production of CO2 and H2O. Here also CO2 and H2O. Here ethanol and CO2. But here in case of lactic acid fermentation, it is only lactic acid. So our correct option is lactic fermentation. Yes. Next 15th one. Which statement is wrong for Krebs cycle? So there is a one point in cycle where FADH2 is reduced to FAD plus is reduced to FADH2, yes, succinate to fumarate. Then during conversion of succinyl-CoA to succinic acid, GTP is synthesized, that is also correct. The incorrect we need to, wrong one we need to select. The cycle starts with condensation of acetyl group or acetyl-CoA with pyruvic acid. This is wrong. It's not pyruvic acid, it is oxaloacetic acid. So here it is OAA. So our option is C. Then phosphoenol pyruvate PEP is the primary CO2 acceptor in case of C4 plants, because C4 plants, in case of C4 plants, they have mesophyll cell. Yes, Niharika, 15C is correct answer. Then 16th one. So mesophilic cells will be rich in PPKs, and because of that, they'll accept this PAP is primary carbon dioxide acceptor. Then next is 17th one. 17th. So which one of the following growth regulator is known as stress hormones? Last two questions. Which one of the following known as following hormone is known as stress hormone? ABA, ethylene, gibberellic acid, or indole acetic acid, IAA. Correct answer is ABA. Abscisic acid is also known as stress hormone. Then last, maybe last 18th question. Fruit and leaf drop at early stages can be prevented. Prevention we need to discuss can be prevented by the application of ethylene, oxine, gibberellic acid or cytokinase. Yes, Niharika, 17th A is correct. Now 18th question, I'm waiting for your answer, Niharika. As you are answering, fruit and leaf drop at early stages can be prevented by application of these. Ethylene, oxine, gibberellic acid or cytokinase. Quick, quick. Yes, our answer is 18th B, oxine. Last few questions for your homework. So for before that, let me tell you that we have a Telegram channel also. You can join us on Telegram. And these are your homework questions. Which enzyme breaks down the fructose 1,6-bisphosphate, 1,6-diphosphate? Yes, come on, answer these questions. And these are your homework questions. After the class, you can post your answer 1 ABCD, 2 ABCD, 3 ABCD like that. Yes, next question. Pyruvate dehydrogenase is used in converting what? Third question. Last electron acceptor in respiration is what? Last electron acceptor. And then when, or you can say terminal electron acceptor. Then fourth question, when 2 pyruvic acid forms 2 lactic acid by anaerobic respiration, then it results into 1 ATP lost, 6 ATP lost, 3 ATP lost, or none of these. Then next question, inside an active mitochondria, most electrons flow which pathway? Pathway of electron, you need to decide and answer these questions. I'll be waiting for your comments after the session. Yes, everyone. Yes, if you like this session and if you like me to, if you want me to take more these type of sessions, do not forget to like, share, and subscribe. And do not forget to subscribe our channel and press the bell icon for notifications, guys. So this is Spark Academy, all the results of 2022. So now 2023, we are waiting for NEET 2023. Altogether, May 7th. Yes, Niharika, I know there will be some gap, but I don't know why this much time it is taking. So I'll be waiting next time onwards more than 5 minutes. So see, if I wait for 5 minutes at each question, then it will be completely, see, that's why I was not waiting. It's okay, Niharika? As you answered, I can see your answer, correct answer here. It's okay. So these are our ranks, some ranks of JEE and why admissions are open right now, and you can be here in our next PPT after May 7th. Okay, when you will be, when I can see all of you wearing a white apron with stethoscope, and you may be one of these person. Thank you so much, guys, and do not forget to like, share, and subscribe. And I'll be waiting for your comments below of your homework questions. Thank you so much, everyone.